Hi, everybody. I can't actually be there today, but I want to ensure that you go through and do this lab correctly. So I want to run through it and explain everything that you're going to do about DNA microarray so that when you do actually go and do it, that it's going to hopefully give you good possible results. So coming at you from my office, what you're going to need is your handout from last time. So this is part 2 where you made your predictions about what you think is going to happen. You had to look at the 6 different genes and what they do, and then predict a person with lung cancer, for example. Would it make sense that that gene would get turned up, turned down, stay the same, or it's just never turned on? And then down here is going to be where you record your results. And then there's going to be some follow-up questions. So you need that. The other thing you're going to need is the white procedure paper that tells you exactly what it is that you're going to be doing. I've set out everything you should possibly need, so that part's taken care of. There's going to be stations. You're going to work in partners. There's more than enough materials to just work with one other person. That does not mean work in a group of 6 where one person does all of the work. Partners. Okay, so what you're going to need. First thing you're going to do is obtain the following things. So you're going to need a card, and it will look like this. It'll either say A, B, C, D, or E, F, G, H. It really doesn't matter. You just need one of them. You'll notice on the bottom that there's liquid in there. This is kind of like the wells that you're going to use. This is your patient DNA, but not all of it is probably at the bottom. So what you need to do is literally like slap it on the table to get the liquid down to the bottom as best you can. So make sure you do that piece. You need that. You're also going to need your card, and it looks something like this. This is essentially our DNA microarray chip. This is where we're going to eventually see all the different colors, but as you can see, it's a piece of paper, not really a chip, but that's okay. Along the side, it has 4 different patients that we are going to be investigating to see what their gene expression is like. They were explained in the gold packet, so if you go to part 2 in the gold packet, it does talk about those 4 people. Across the top, we have a couple of different things. The first 4, as it explains to you in the procedure, are really our controls. So as it describes on the bottom part of your procedural page right here, sample 1 is normal expression. So that's what it should look like if everything is normal. Lane 2 or sample 2 is going to be what it should look like if expression goes up. And hopefully you've learned that when expression goes up, you're going to be expecting a red color. Expression goes down is the third, that controlled. Those should all be green. And then the 4th one should be blank, meaning that it's not on at all. So that one should be kind of black or you don't see. And as I failed to mention earlier, the normal expression means that it should be yellow. So those 4 are kind of your controls. If you don't see those colors, that's a problem. If you do see those colors, then most likely you did everything correctly. And then from there, lanes 5 through 9 are the actual genes that you were testing that you made those predictions on. So that's what's happening in this chart and how you'll see those colors later. So in terms of what you're doing for the actual procedure, that's what the back side of your procedural page is. Read the directions, please. So you're going to use a micropipetter. That's the pink guy. You're going to set it to 5. It says 5 microliters. And you're going to have a couple of different solutions at your station. So the first one you're going to use is called equilibrium buffer. I'm going to pipette it out into little tiny tubes for you and have them labeled, but it's going to be the one that is blue. Equilibrium buffer in this case is going to be blue. So what you do is you take 5 of that and you're going to drop it onto each and every one of these dots. So, 4 times 9, that many, 36 dots on each one in the center. Then, once you've done that, step 4 says place it in the incubator for 5 minutes. The incubator should be there. It's along the side towards the back of the room. It's already on preset to 37 degrees. You're going to put it in there for 5 minutes. But before you put it in there, please make sure you write your initials somewhere on this card so that you'll know which one is yours compared to everybody else's. Once the 5 minutes is up, you're going to take this out. You are going to get a fresh tip. You can use the same tip on the previous round because it's just the same solution every time. For here, you're going to take control DNA, which in this case is going to be green. It's kind of hard to tell, but it's this weird green color. You're going to take this. You're going to get 5 microliters in here. This is weird now. Pay attention. 5 microliters will be in your pipette. You're going to come to the tray. You're literally going to punch through. It's kind of like a punch system here. You're going to punch through and pipette up and down. The goal is to mix that green solution that you just had with what is already in that well. So you're going to punch through the foil that's on top, kind of mix it up and down, and then take 5 of that. So you can use the same tip, just mix it up and down and then suck up so you have 5 in the tip and put it on the corresponding well on the sheet of paper. So A1 here, this very corner one, is going to correspond to the corner one over here. And then you're going to discard the tip, get a fresh tip, and repeat the whole process 35 more times. So you're going to take 5 of your control, punch through the foil, mix it up on the next well, and then put the 5 of that on the card. Repeat, repeat, repeat. So there's the exact same number of these guys as there are these guys. So you just have to make sure that you're keeping up with the corresponding well. So once you've done that, then you're going to put it back in the incubator for another 5 minutes. It just basically dries it out and allows the chemicals to work together. You're almost done. The last step is hybridization. And what you should have learned is that hybridization is when the sample from the microarray, so that's already pre-laid with DNA kind of, and you add in your samples, patient sample DNA. In this case, it's going to hybridize, or wherever it's complementary in the well, it's going to then stick to whatever you have for your patient. So if that gene is on in normal cells, it's going to stick. If it's on in cancer cells, it's going to stick, and you'll be able to see the ratio of before and after. So in order to do that, the last thing you're going to do is add 5 microliters of hybridization buffer. And this one is going to be the clear one, and it'll be in a tube marked HB for hybridization buffer. Put it in the incubator one last time for 5 minutes, and then you're ready to see it. So at that point, it's done. There's a total of 15-minute wait period in the incubator, give or take a couple of minutes to do the different samples. I'd say the whole thing can be done in about 30 minutes or less. But the last step is to visualize it. So you're going to need this guy. This is the UV box. So it should be plugged in. Just make sure that this guy is turned on. And then we did this for the same as the gel reading. The chemicals that we used in this case are UV fluorescent. So you're going to lift this up, put your card down, close it, and it should glow. And then you can go ahead and take a picture of it. The idea then is that you take your student response sheet here and you're going to actually put in or fill in the colors. So use colored pencils, whatever you need to do. The idea is that you note whether it is red, green, yellow, or blank. And it might be to a different degree, like there might be 2 greens, but one is way more intense than the other. So please make sure that you note intensity as well, because that gives an indication for how high the gene is turned up. Okay, so once you do all of that magic, then you can go through, there's a little chart here for you to interpret the colors of did it get turned up, turned down, what color does that mean, and a couple of follow-up questions as to kind of which genes should we now investigate further. Once the lab piece is done, you should be able to finish everything from 3.1.4. Next class, we're going to move on to 3.1.5. So you need to have this data. I'll go over it with you, but you need to have all of this done. I am entrusting you to get this lab completed and get a picture of your beautiful microarrays in my absence. Please tell me I can count on you, and good luck.